Yes. There we go. That way, everybody can go back if you want and take a look-see at the information. So we're going to cover what to do with your bees getting ready for winter. So I'm going to share my screen here. Come on. There it goes. So last call before winter, everyone. Believe it or not, that picture is my backyard last year. And so get ready, everybody. We've had that much snow on top of our beehives the last 2 years in a row. Yay! So I'm going to start with setup for winter. So what should you pay attention to right now to hopefully provide success for your colonies over the winter? And remember last month, if you were here, we talked about Varroa management. So if you haven't checked your mite level yet, please do. And if it's above 2%, please do something about it. I'm already reading on Facebook about how my bees absconded. That I had this huge colony and now all of a sudden they're gone. Well, they didn't probably abscond, except they probably left one at a time to go off and die because they had mites, and the mites made lots of viruses proliferate in the colony, and the bees fly away to die so that the colony doesn't have to deal with their corpses. So that's what happens to colonies this time of year. They just disappear, and it's all due to mites and the viruses that are vectored. So we want to have, of course, strong colonies with big populations. So if you have more than one colony and one or both of them are kind of small, now's a really good time to do a newspaper combine and make one big colony so that they'll have lots of bees going into the winter. Having a young, high quality queen always helps. I tease Kathy every year because she hates killing her queens. Kathy, you're smiling because you knew I was going to say this. She hates killing those old queens, and sometimes she'll have like a 3-year-old queen going into the winter, and then the next spring she'll say, "Oh, damn, they died." I'm like, "Well, of course they did. The queen didn't make it." She's like, "Oh, I don't like to kill them." So that is true. It can be a problem. So you want to have adequate honey stores. I'm going to talk more about that in a little bit. And you want it located above the bees and around the sides. You also, it's not a bad idea to have emergency food if necessary. And then we were debating upper entrances before you all joined. I'm a proponent of upper entrances in the Northwest anyway, because we have all that warm, moist air that makes it hard for the bees, because if it doesn't have anywhere to vent out the top, then it tends to rain back down on them and kills them. And then because we do have frequent windstorms, it's a really good idea to put a weight on top of your colonies. I use old scuba weights. You can use bricks, anything that's heavy, or a tie down ratchet strap works really well if you're in a high wind area. And then install an entrance reducer. I'll tell you what that looks like in a little bit here. And if you live on a farm and the colonies are down low where you might have mice getting in, consider a mouse guard. There's nothing grosser than cleaning out a big mouse nest in the bottom of your colony. In the springtime, I've done it. It's nasty. You don't want to do it. Add a mouse guard. When in doubt, add a mouse guard, I say. So what does a fall inspection look like? I mentioned this before. Consider replacing your queen if she's old. Choose a warm afternoon and move all your frames with brood into the bottom box or boxes if you're running mediums, and they won't all fit in one box. And then bracket honey around it. I'll show you what that looks like on a separate slide here. If it's warm outside, that's great, above 55 degrees, although you do need to work quickly this time of year, especially if you have multiple colonies, because it can induce robbing. And so you don't want to have the boxes open too long. You do want to reduce your boxes. So if you have not already taken your extra honey super and queen excluder off, now's the time to take care of that. And while you're out there working, an extra lid or a piece of plywood, something to just cover up the top so that the robbing bees can't smell it. And that's a good idea. I mentioned uniting smaller colonies before. So the last thing here is, what do you see on this frame? Eric Luthi shared this great frame with me last fall, and I asked his permission to share it with you guys. So what do you see that might be unusual this time of year? Kathy, you're on mute.
It looks like they might be honeybound.
Maybe.
And it's,
There's a queen cup in there.
There is. There's
Oh.
two.
yeah, I can't see that.
Two teacher cells. So if your bees are superseding this time of year, it's a really good idea to look for your queen and maybe replace her, or maybe the bees, maybe she's already dead and the bees are trying to replace her and it's kind of too late at this point, because there aren't very many drones left, and these may or may not have eggs in them, but they are definitely deciding that they probably don't like this. What's missing by the green arrow that should be there?
Pollen interface?
Yeah, exactly. There's no pollen on this frame. So unless there's pollen on other frames, then this colony is probably not going to be raising more brood. This frame has a lot of capped brood, so I'm guessing that they just exhausted all the pollen off of this frame and that there's more somewhere else. But if your bees are not laying more eggs right now, they should be making winter bees. And so if they're not laying eggs right now, you need to get some pollen patty in there so that that'll help give them a boost so that you'll have enough winter bees. Kathy.
I just wanted to mention that those little circles around those queen cups, those are just cells, and bees are always prepared, and you'll find them throughout the hive, but until there's an egg in, they're not superseding.
Yeah, so Eric said, I think last year, that these did have eggs in them, but that's a really good point. Nice that this colony still has at least one drone left in the yellow circle. So I haven't seen drones in mine for a couple of weeks now. Kid, do you have drones still?
I do.
Kathy still has drones.
I do. Couple of hives have tons of them. As of a week ago, I've left everybody alone because I treated late, but I'm treating.
Okay, great. So there's hope for my one virgin queen. All right, I promised that I would show you guys what it looks like. So this is just a real simple schematic. The yellow is honey. So this is what you want your colony to look like as you do your last inspection of the fall. You want all the honey that you have up top. You want a frame or two of honey on the bottom if you have extra. And then you want to put the brood and any eggs in the center on the bottom. Capped brood goes next to that, and then any nectar or pollen right next to that. So everybody's boxes will be slightly different, but this is the general idea of what you want your colony to look like for your last inspection of the year. All right, and fall feeding. Now, if your colony does not have 70 pounds of honey in it, then you're going to want to feed. And you're going to want to feed 2-to-1 syrup. That's 2 parts sugar to 1 part water. And you do need to heat the water to make that all get dissolved in there. How much is 70 pounds of honey? Kathy picks it up on the back, and well, she tries to pick up the back, and if she can't budget, then that's plenty. I can usually lift 70 pounds off the back, but just grab the back with your hand and just lift up and see if you can pick your colony up, tilt it up. So 1 frame capped of medium frames weighs 3 to 4 pounds. And then a capped frame of a deep frame, that's 8 to 10 pounds. So you can guesstimate as you're going through your colony doing your inspection to see, all right, well, a honey arch might be like, I don't know, half a frame full. Maybe that's 5 pounds on a deep. Guesstimate that way to see if you're up to 70 pounds. If they're short now, make sure that you're feeding them while it's still warm enough. They will not take the syrup once it gets down below 55 degrees Fahrenheit average temps during the day. They'll stop taking it. And once that happens, if you have extra left, don't worry about that. Just stick it in a milk jug or something and freeze it for the winter, and then you can dilute it for the spring. So also I have on here, use internal feeders if you can, because it does discourage robbing. If you have those Boardman feeders that slide into the front, you're going to feed all your neighbors' bees, and it'll say, hey, there's food over here, come get it. And so you'll tend to create a robbing frenzy if that happens. If you only have a Boardman feeder, go ahead and put it on top of your inner cover and then put an empty box around it, put your lid on top of that. That way you create your own internal feeder. And then once it gets colder, usually by the middle to late part of October, the bees stop taking the syrup. And if they're still light, or if you want to have just an emergency store up there, you can add a shim. I've got a picture of that in a little bit, and start feeding dry white sugar. And then right now, I mentioned earlier, if you're short on brood, go ahead and add pollen patties, or if you don't see any pollen coming in, make sure that you add pollen patties till probably the beginning, like mid to late October. And then around here, most of us do use moisture quilts and shims for feeding. So you see, this is called mountain camp feeding in one of my colonies. I just put a sheet of newspaper right on top of the top frames, and then I pour sugar on top of that. And this sugar is lumpy because it's been in there for a little while, and it doesn't go all the way across because they'd already built comb and put honey in part of it. So I just poured it up against the honey frames that they had already instead of tearing that out and left that for the winter. So that's the shim that I have. If you look on the right-hand side where all the bees are on the shim, that's a 5/8 inch hole that I have for the upper entrance. And then on top of that is moisture quilt. And so I just use scrap lumber for this. And I use, if you're making it, you can use extra medium box that you have, and I use window screen on the bottom, the aluminum window screening. You can use 1/8 inch hardware cloth if you want, but it's more expensive. And with nice big vent holes on the side. And then I use rabbit shavings. Whatever the cheapest pine shaving is that they have at the pet store is what I toss in there. And if it gets a little bit wet in the winter, you can just take the wet stuff out and pour nice fresh dry stuff in there. And then in the spring, I dump it all in a sack and burn it in my smoker all summer. So you see on the bottom right-hand side, the colony that's all put together. So you've got the 2 deep boxes or 3 mediums, and then the shim with the sugar feed in the top, and then the moisture quilt on top of that. So here's what my winter hives have looked like over the years. So you've got the 2 boxes. Notice I say there's no need to wrap hives here. It doesn't get that cold. And if you do your last inspection in late October-ish, then they have plenty of time to propolize all the cracks. And if you use a ratchet strap, that'll really compress everything all together and keep the cracks from leaking any sideways rain in there. I do use 1 inch rigid insulation in my lids. Seems to really help keep the heat in and keep the moisture from raining back down on top. And then I do place my sticky boards back in the screen bottoms. Some people do, some people don't. It's the wet that kills our bees, it's not the cold. So the cold might create starvation if they don't have enough food, but the cold is not going to kill them. So one year actually at my cabin, I forgot to put my sticky boards in, and guess what? The bees were just fine next spring when I went back over there. So I don't know what the right thing is on the sticky boards on the bottom, but I'll tell you what, they are kind of nice to be able to pull those out in the winter, and you look down on the bottom to see where the debris is, and then you know where your bees are. And I know, and I acknowledge again that that was a lot of stuff. So I think we probably have a little bit of time for questions if you guys have any. I see there's some stuff in the chat.
Yeah, we have one question from Bill in a chat.
Is there any problem with feeding winter syrup simultaneously to do a mite treatment, i.e. Apogard? So the package insert on ApiGuard says not to feed. And so does the insert for Formic Pro and Mite Away Quick Strips, but it doesn't say why. So when we were in the bee class, one of my colleagues called each of the companies and asked, and they said that they had done trials and that it wasn't statistically significant, but there was a trend in both of their trials towards lower winter survival in large studies of colonies that were fed the same time as they were treated, but it wasn't statistically significant. So if your bees are desperate for food and you need to put ApiGuard on, I'd say go ahead and do it. I do it most of the time too. Or I'll stagger it. I'll put the ApiGuard on 1 week and then I'll feed the next. And then I'll put the second treatment of ApiGuard on and then I'll feed after that. Any other questions?
I see 9 questions in the chat.
All right, I made the chat bigger.
There is one about robbing screens and whether we could put
Same.
extracted frames in the same general area, I'm assuming somewhere around the hives to be cleaned by the bees.
Okay, those are both great questions. So if Hive has robbing screens on, do you have a problem with putting on? No. So when I take my extracted frames off, I put them back on top of on a weaker colony, or one that doesn't have as much food. And I just make sure that their entrances are small. And then I go ahead and let them have that honey, and it's all cleaned up in a day or two. And I personally haven't had a problem doing that, but like I said, I make the entrances small. So you should be okay. Medium-sized colony. If you don't have another colony to combine, are there other suggestions? Yes, hope for the best. So flippant answer, but you probably noticed on one of the those 4 colonies of mine that I have together. One winter I had 2. They were all in double deeps, and the next winter 2 were in single deeps and 2 were in double deeps. So if you have a 8 to 10 framer, that's obviously 1 box. So the best you can do is make sure they're healthy, that you're putting pollen patties on right now to try to give them a little bit of a boost, and then just make sure that they've got enough food for the winter. And read Rusty Burlew's blog about winter feeding. A lot of people say that they don't want to get in and feed them in the winter. We'll talk about this, in January, but it's better to crack the lid open and feed them and let them get a little cold than to let them starve. Amy, is your helper raising his question? Or raising his hand for a question?
So is it ever so okay to take honey from a new hive?
Yes, it is okay to take honey from a new hive. That's a great question. So long as your bees have more than 70 pounds, if they have extra, you can have that.
That's like 2 of you. We accidentally caught a wild hive this year. So this is our first round of beekeeping in a long time. So thank you.
You bet. Swarms make a lot of wax. So those are great catches.
Thank you.
Is the honey good? Have you tried it? Or did you let the octopus eat it all?
An octopus.
We haven't tried any yet because they're so new, but he's very excited and he got a beekeeper suit his size for his birthday. So we're very
And I got a robot.
and a rope. Yes. Thank you.
That's awesome. What about moisture boards or quilts better? So Karina asks, moisture boards or moisture quilts? They both work, and Kathy's used moisture boards for years and years. They work great. I never bought any, and I went quilts and they work great too. So whichever you prefer. Is it possible to get a copy of my presentation? So it'll be taped. or is being taped. So you can watch the presentation on YouTube. Where do you put the 1-inch rigid lid? Easy for me to say. Where do you put the insulation? So I have telescoping lids, and I just put the insulation right in the lid. So the inner cover goes down, and then the insulation on top of that, and then the telescoping lid. I usually cut mine so they're tight enough that they cram up in the lid and don't come out. And I leave mine on all year round. Kathy.
Oh, I wanted to tell Amy's helper, whenever I have new beekeepers and I'm helping them go through the hive, I always tell them to stick their finger in and take a taste. The bees will clean it up and they don't mind if you have a little taste. So get yourself a taste of that honey.
Thank you.
You're welcome.
Yeah. Karina asks, should we take the robbing screens off before winter, or should we just keep them on all year round? Either way, whichever you prefer. I've left them on all winter. I've taken them off and put entrance reducers in. It really, I don't think it matters. How about if you treated, how long? Oh, so I think Melissa's asking, what if I've treated? How long until I can get honey? So it depends on what you used. So that's why most people don't extract honey from the frames they use in their brood boxes, because those are the ones that get treated. So depending on what you use, it's wax is really lipophilic. So like Apivar goes right in the wax and it never really comes out. And the bees, of course, the bees bring back tons of pesticides and stuff all year round. That's why most of us rotate our wax out. So usually what I do is I pull my supers off and then treat, but like Formic Pro you can use while your supers are on. So that's really the only thing that's approved to use while your super's on. If you used anything else, then you should take your supers off before you use it, or use it before you put your supers on. Does that, Melissa, does that answer your question?
Yes, we're new, so we're just learning.
Yeah, you know what? I'm learning too.
Okay.
It's a constant learning about bees. Just when you think you've figured them out, you haven't.
I have another question about the rigid insulation.
Sure.
Are you talking about like the pink foam stuff that's fairly rigid? Because I understand that sometimes the bees will just rip that apart, like eat it not eat it, but like.
Yeah, they eat the white stuff too, the styrofoam stuff. And,
yeah.
And as soon as they start eating a hole in it, I flip it over so that they have to start over. You can use either the pink stuff or I just use the white rigid insulation. It's
Okay.
white on one side and silver on the other. Do I shrink wrap the entrance for the winter? No. So it's the moisture that wreaks havoc in our colonies. So it's best to have ventilation going. And so I don't recommend wrapping our colonies here because it just makes more water on the inside. Looks like you leave an entrance at the top as well for the winter. Right, I've got that 5/8 inch hole in the top. And so what happens when the bees die in the winter, because some of them do, they fall down on the bottom. And so they can't always get out if they, you only have a bottom entrance. Plus, if the bees are at the top and they're all clustered together, and one of them has to go to the bathrooms, they won't break cluster and go down and out. But they will go out. Even when there was snow on top of my colonies, when it warmed up in the afternoon, bees would go out and take a poop and then come back. So that upper entrance makes it a lot easier for them to do that. And it also vents out because that hot, moist air goes up, right? And so it goes up into the chips or into the moisture board, and then it also vents right out that hole. That way it doesn't condense and then rain back down on them.
Okay, I think we need to pull away from these questions, which I have a couple too, but I'm gonna hold my thoughts. Because it's time to move forward. We have an action-packed evening. We have Tom Seeley is on the East Coast, so we have to respect keeping our time frame quick. And to that point, I will not be talking for long. I'm simply going to say welcome to PSBA, and I'm so glad you're here for our September meeting, and let you know that we're working on setting up breakout meetings in October. We'll have more of a social meeting where we can talk to each other. And that's really what we talked about at the board meeting, how much we miss talking bees. So we'll have several breakout rooms and you can select which room to go to, and we'll have people, mentors and leaders, and intermediate rooms won't really be leaders. They'll just be there to back things up. And so that should be fun. I hope everyone agrees. And with that, I'd like to introduce Dr. Seeley. Are you there, Tom?
Yes, I am.
Okay, good. I'm so glad. And the introduction that I put in my president's newsletter is extensive because that is the nature of Tom Seeley. He has an amazing repertoire and quite a reputation among beekeepers. A couple people that I told that he was going to be speaking said, "Oh, I'm reading his book right now." I think Honeybee Democracy might be the one that people resonate with most. His most recent book is The Lives of Bees, which came out a couple of years ago. And I just love the direction that he takes his research. So with that, I would like to just pass the baton. And I also want to explain that what we will do is try and address the chat. That's with so many people on the call, we'll really be leading on that chat so that we can keep. Tom wants to take questions, but it isn't. That will have someone able to answer many of the questions, and then some will feed into Tom. So Kathy and Magali, maybe you both can kind of be on the chat and work together to keep things rolling, and then ask. Does that sound right, Tom? You'd like to handle questions at the end, if that works, and not interrupt your speech, or
I will. I changed my plan a little bit. I'm going to make, I think, 3 breaks partway through.
That's right.
And ask do maybe 3 or 4 questions at each one.
I'm sorry, you told me that. That's a beautiful plan. Okay, thank you very much, everyone, and welcome, Tom.
Okay, I'll share my screen, and there we go. There. Good. Does that look good at your end, coming through?
Yes.
Great. Okay, good. I'm just going to adjust this a little. I'm really, it was a pleasure to listen to the discussion and hear the advice from Tracy about wintering. It's you guys have a different world. It's got a lot more moisture in your air. Our winters are cold and dry. So all beekeeping is local, and that was hammered home in hearing the discussion tonight. So what I'm going to share with you tonight is a topic that's I think you will find useful, and I'll explain why that is. We're going to be looking at how bees live in nature or in the wild when we have nothing to do with their lives. And I want to start by just making the point that it's a huge difference between how they live in the wild and how they live under management, at least under commercial beekeeping. What we see on the left here is a photograph of one of the blueberry barrens up here in Maine, where I am. And you can see electric fence to keep the bears out, the black bears, from destroying the hives. But the main point is there's a lot of colonies crowded together there, whereas we'll see in the wild, it's a very different. Colonies are widely spaced, for example. So here's the basic 2 parts of the talk. How do honeybee colonies live in the wild? Question 1, and question 2, how are these wild colonies surviving with Varroa because they all have Varroa, but without any treatments from a beekeeper or beekeepers. And I will be sharing with you what I've learned over the years about colonies living in the forests outside my hometown of Ithaca, New York. I'll show you where that is and give you a feeling for what the habitat is like. And these are the key questions. What are the bees' homes like in the wild? How abundant are they? Do they have Varroa? And the answer is yes. And how are they surviving without any attention from, do they have good genes? And also, do they have good behaviors or / lifestyle? And the answer to both of those is yes, and we'll see the details. A little bit of information about location. I see that we're pretty much at almost the same latitude, and we both have cold winters. And so I think this talk will have good relevance, even though there are, of course, differences too. So I'm living in a small city called Ithaca, New York, and it's about a 5-hour drive from New York City. So people say New York, they think of New York City, but most of New York is actually quite rural, as we shall see. The vegetation here is primarily deciduous forest, few conifers, spruces, and white pines. And our climate is a continental climate, really hot summers and really cold winters. This is my house, and that's not unusual, that much snow. Well, part one of this talk is, what are their homes like, these wild colonies? Well, they are living here in Ithaca, New York, in buildings, people's farmhouses and hunting cabins, things like that, but mostly in tree cavities. And here are the two examples. And just an entrance here and an entrance there. We'll be looking inside those cavities shortly. Now let's talk a little bit about the characteristics of their nest sites. Most of the wild colonies have an entrance that's high off the ground. The average height is 29 feet. And we'll come back to why that is. It has something to do, give you some hints. It has something to do with black bears, and it has a lot to do with snow. So they're high up, and that's me, and that's a nest entrance up there. In terms of the size, most of the entrances are small, 2 to 3 square inch openings. So that's about a quarter of the size of the entrance that comes on a manufactured 10-frame Langstroth hive. Relatively small. See why that is too. What's the size of their nest cavity? Again, things are scaled down. It's about the size of one deep 10-frame Langstroth hive body. This is a distribution of the nest cavities that I measured when I collected 28 wild colonies, or no, 23, forget what the number is. And you can see there's a broad range from less than 20 liters, which is half of a 10-frame hive body, out to a gigantic one, which was way up in the top of a cavity of a big beech tree. The nest was small, and they were way up in the top of this cavity, so it was huge, but most of them are in something in the range of a little bit less than 1 super or a bit more. So that's a really, as we'll see, that's a huge difference makes a huge difference, as I'm sure you can imagine, in how a colony lives. It means they can swarm often. What about this cavity structure and shape? Well, here's this sugar maple tree I cut down and cut out the section with the nest in it and split it open. And here's what it reveals, which is typical. It is a thick-walled cavity. Typically, the walls are several inches thick, and it's tall in the shape of a tree. And this nest is an unusual one, and the entrance is near the top of the nest. Almost always in the bottom, almost always in nature, it's in the bottom third of the nest cavity. So this shape will come back to it. The bees have adapted well to this, and it's got this, but you can see the layout is typical. There's a brood nest in the bottom, and the honeycombs are up top. You may be able to pick out too that there's a patch of drone comb down here. Total comb area relatively small. It's about 11 square feet or 1 square meter in these wild nests. And that's very different from managed colonies. I always like this advert from one of the, I guess it's Dadant bee supply catalog. The best and most complete wooden kit. It's a gigantic, it makes for a gigantic home for the bees, which is of course what a beekeeper wants. They want a big, strong colony with lots of storage space for honey. But it is, that's probably good for the beekeeper. It's sometimes, and we'll see, it can make some challenges for the bees, especially dealing with Varroa with so much brood. So in the wild, it's a much smaller operation. It's about a third the size. What about the sizes of their cells? There's a lot of people are interested in this, whether the colonies in the wild have small cells. Well, they're slightly smaller. Where I live, they're 5.4. If you build a comb on standard foundation, it's 5.35 in the millimeters in the wall to wall in the wild colonies nests. They're not small cells. They are apparently in the wild colonies in the Southwest, but that's because those bees are very much have the genetics of Apis mellifera scutellata, a different race of bee. I'll come back to what the races of what racial mix we have in the Northeast. Drone comb, 17% of the comb area in a wild colony's nest is devoted to drones. You might think, why do the bees want to build so much drone comb and rear so many drones? Well, when you think about it, drones are part of how a colony passes on its genes. And if a colony didn't have drone comb and didn't rear drones, it would not be genetically very successful. So these colonies are making sure they've got the means to produce drones. Propolis. Interesting discussion already tonight about Propolis collection. In the wild, they go wild for propolis. They will coat the bottom, the floor of the nest here. This was a nest cavity that had been occupied by a squirrel. You can see the hickory nuts that's left behind by the squirrel. They were all plastered like linoleum in the bottom of the cavity. And here's a photograph of the wall of one of these tree cavities. Now, this is what it looks like normally over here. Everything's coated. I just chipped off the propolis here so you could see what's beneath it. And I think this reveals also why the bees are putting so much propolis on the walls. Imagine trying to control microbes or attach combs to this friable, decaying wood on the sides of a cavity. They rip off as much of that loose wood as they can, and then they plaster it with propolis. The whole story of Propolis Collection is a very fascinating one. I don't have time to go into it today, but it's a special group of workers that are focused on propolis collecting and processing. And when the bee comes in with a load of propolis, she doesn't pass it off the way she does her nectar in the entrance. She goes up where the propolis is being used, and then she stands around until one of the bees that's working the propolis chews it off her pollen baskets and makes use of it. It's a neat little part of the life of a colony. So let's think about what some of the consequences are of the way they nest in the wild with thick walls, small comb areas, lots of drone cells and much propolis. Are the nests well insulated? Yes, they are. They're in these thick walls, thick-walled cavities. Small brood nests and small colonies? Yes, that's what's going on in the wild. Much colony reproduction through swarming and drone rearing? Yes, colonies invest a lot in reproduction in the wild. Long live queens? I don't know. I'm not able to track that in wild colonies. I can track it in simulated wild colonies, and there, yes, the colonies, it's not unusual to have queens live 3 years. Less disease? The answer is yes, and especially, as we shall see, less stress from Varroa, and it has a lot to do with these small colonies and small and frequent swarming. So here's my first break on questions, and what if people have questions on what I've shown so far, please put that in the chat, and I think Magali or somebody else is going to read a few questions, and I'll take those now. I don't want to and so I want to make sure what I've said so far is clear.
I'm happy to do that. Everyone, feel free to put any question you might have in the chat. Here's one. Is there a way to simulate small colony size and frequent swarming and still harvest honey? I'm assuming in managed colonies.
Yeah, there is. And I'll talk a little bit about that. The main thing is you just scale back your expectations of the honey. For example, when I want to manage bees in what I would call a more natural way, I put on just one shallow honey super. And then, so I'm not taking a lot of their honey. And I would be giving them 2 deep hive bodies. I use 8 frame equipment. So 2 deep hive bodies with 8 frame equipment and then a shallow honey super. It's a little lighter hit on the colony.
Thank you.
Okay.
And I have the question, have you found nests in fallen trees and how different are they?
Oh, what kind of trees?
Fallen trees, dead trees on the ground?
Yes, I have, but it's the bees have, it's because the tree, I occasionally do find a tree where I've been following it with a wild colony nesting high up in it, and then it falls over for whatever reason. It's been weakened at the bottom. And I see that. And the bears really like it when those trees fall over with the bees in them. But they try to rip open the nests, and sometimes they do, and sometimes they can't accomplish that. But it's not a common phenomenon, but it does happen. Bees stay there.
That's really cool. And then I had another question, which was, what particular race do you tend to see in the wild?
I'll come back to that question. It's a hybrid of Apis mellifera mellifera, Carnica, Ligustica, and a little bit of Scutellata now.
Cool.
Okay.
If you can take one more question, there is one which is what is the best way to recreate those thick walls that you see?
The way I'm doing it is I take a standard hive and I put 1.5 inches of polystyrene foam on the outside all the way around. It's a bit of work because then you have to put a layer of wood outside of that to put handles on, but that's what I'm doing. Yeah. There are polystyrene hives, which I use a few of those, and the bees work very well in our climate, our cool, very cold climate. Those have good insulation too. In fact, they work really well. I don't like, the only drawback to them is they're plastic. So.
And not very environmentally friendly. We have some more questions, but I don't know if you want to take them.
I think I better move on and we can come back to those if possible. Okay. So I'll close look at the chat. Okay. So we've looked at question 1, what are their homes like? Question 2, how abundant are these wild colonies? And many people have thought they're all gone, so we'll address that too. Well, here's the place to address this question of abundance. I'm going to report on what I find in a forest that Cornell University owns called the Arnott Forest. And I started my work of following the wild colonies in this forest back in 1978. That's the year I got my PhD. And so I've been tracking it for over 30 years now. And over the years in this whole area, this part of the world, it's been getting wilder and wilder. The forests have grown back in more and more. Black bears have moved in, ravens and fishers, and the wild colonies have been here the whole time too, as we'll see. Though they did get a hit when Varroa moved in. Here's a view of what this part of the world looks like. It's not good agricultural land. And so it was occupied by farms after in the 1800s, but people realized this stinks and they moved out. The farms are gone. You might wonder looking across that forest view, how on earth do you find colonies in those forests? I do it by bee hunting or bee lining. This is an old craft, which is also much fun. You go to a patch of flowers. Here's some fireweed. You find some honeybees on the fireweed or whatever, the roses. You capture them in a little box called a bee box, and then you introduce them to a dish of sugar water, I use an old comb, put sugar syrup in it, And then you see the direction that the bees are flying home as they go home on their beeline. And then you work your way down the beeline by moving your feeding station about 100 yards or so, step by step, till you get to their home. And that's a wonderful experience, and it's a great compliment. Being a bee hunter is a good compliment to being a beekeeper, because the nice thing about when you're out bee hunting, you're dealing with a small group of bees, and you really can focus on their individuality because you'll mark them with individual paint marks. Whereas when you open a hive, it's very impersonal. You're just dealing with masses of bees. So I encourage people to give bee hunting or beelining a whirl. Now, to address this, here's this question. How many colonies live in the Arnott forest? I've measured it 3 times, 1978, 2002, 2011. And I do this by just bee hunting around. I can cover about half the forest in the time period that I have in August and September. And you can see the numbers are basically the same all 3 times, about 9 colonies. And if you know the area of the forest and you estimate that I'm only finding half the colonies, it works out that I keep finding about 2.5 wild colonies per square mile. So that's what and I want to stress, this forest is far from any managed beekeepers. The nearest apiary is 5.2 kilometers away, or 3 miles away. A beekeeper did put an apiary one summer down here, and I was dismayed by that, but the bears wiped it out. So he didn't put the bees there anymore. So that solved that issue. He had an electric fence around it, and I did not unplug his electric fence. The bears just pushed through it, apparently. But one thing that we'll look at shortly is there was heavy mortality when Varroa arrived. Varroa arrived in this part of the world in the early 1990s. I first saw Varroa on my bees in 1993, and I guess it came a little earlier. And as we'll see, the colonies were almost wiped out. 80% of the colonies in the wild were killed off by Varroa, but they've rebounded, as we shall see. So let's pause here again and see if there are questions about the abundance of wild colonies.
I see a bunch of questions.
Okay.
But they are from before. One of them was, because they're smaller, when they swarm, how much of the honey do they end up taking with them because they're such smaller colonies?
Well, well, I would have to just do the quick math on it. There's about 15,000 bees on average in a swarm. Each bee is a full honey stomach, so she's got 40 milligrams. So 40 milligrams times 15,000, that's 600,000 milligrams. So it's about 600 grams, not a lot of honey. That's about 1.5 pounds of honey in a swarm, not a whole lot.
Thank you. There was some impressive math calculation, I want to say that.
I keep a piece of paper on the desk.
Then we had some questions around how long colonies stay in a particular place. And so there was a question around how long do they stay in a given location, or do they actually tend to vacate the premises entirely at some point and then choose a different place?
No, I've never seen that. I've never seen a situation where a nesting site becomes vacated. What I do see is nest sites get vacated, but it's in the winter when they die off. There was one time when I did experience a colony abscond from one of my hives, but it was one of my observation hives, and I had left the lamp too close to the glass, and I overheated the colony. That's the only time I've ever seen a colony just up and leave its home. But so no, I don't see that.
Yeah, that's really good to know. And another question that was related, I don't know how you would have estimated that, but can you tell how old the colonies are when you find a wild colony? And if so, what's the oldest?
Yeah, I can't tell the age of a wild colony when I find it because I just don't know how long it's been there. But what I do is I start keeping track of that site, and I will check the site three times a year, spring, midsummer, and fall, to see if it's survived throughout the summer. And then I'll check it again the next year, and the next year, and the next year. And from that, I learn about how long a site is continuously occupied. And the longest that I've seen a site continuously occupied is 8 years. So there is a good deal of turnover in the sites. The average duration of occupation of a site is about 3 years in the wild.
Yeah, it's very good to know. That is a lot shorter than I would have thought if you consider that they won't abscond. And then I have one additional question that just came in, which is, are there similar wild bee surveys in other regions of the US that you know of?
There was a survey done in Louisiana by the USDA, and they were interested in how many, they had the African bee there, or still do, and they were concerned about what the abundance was. The other area that is like and I caught in 2003 and 4, those 2 summers, I caught 11 swarms, and every colony that moved in had Varroa. So yeah, these colonies out in the wild have Varroa. One of them came in with chalk brood, and that was the only other disease. None of them came down with foul brood, either foul brood. But Varroa, definitely, they've got Varroa. But a curious thing happens with the Varroa. I would monitor the mite drop. I did mite drop counts in these colonies one year, and counted the number with sticky boards and counted how many mites dropped in 48 hours on the sticky board. I do that once a month, and these were colonies that came through the winter, so they were established colonies. And you can see it starts low, starts low, starts low, starts low, goes up in the fall, but it's the late fall. And all of these colonies survive. I think the buildup of the mites is so late, the virus levels apparently don't get high. So that's what I'm finding with the mites. And we'll talk more about these mites in a little bit. So how are they surviving? And this brings us to this question of genes and lifestyle. Do they have good genes? Evidently they do. They have quite good genes for resistance to Varroa destructor. And I can say this with some confidence because I know that in the mid-1990s, the wild colonies, the unmanaged colonies living off all on their own, they experienced very strong selection for resistance to Varroa. And the way I can make that statement is by looking at the change in the genetics of the bees in the wild, living in the wild, before and after the arrival of Varroa destructor in the mid-1990s. Now, to do this, I worked with one of my students who's an evolutionary genetics whiz, and we took advantage of the fact that back in 1977, I collected what are called voucher specimens. Every time I'd find a wild colony, be it in a house or a tree or wherever, I would collect some of the workers with an insect net, and then I collected about 30 of them. I put them as pin specimens in the Cornell Insect Collection. And Sasha, one day he said, "Tom, do you have old bees, bees from old wild colonies? Because the genetic techniques now are such that we could make a very good comparison of those bees, their genetics, to the bees that are living now in and around the Arnott forest and other forests nearby." And I said, "Yeah, Sasha, I've got all these old ones back in the insect collection." So we got those, and we went out and sampled an equal number of colonies from the modern ones in 2011. And what this diagram shows us, which is a diagram of really, it's a family tree of the queens. And so this is the family. The blue refers to the queens from 1977. And I hadn't collected the queens, but you can infer the genetics of the queen from the genetics of the workers. And you can see that at the old colonies, back in 1977, the queens were in many, many different families. I think it was about 23 different lineages in the queens in 1977. But here in the red groups, it's just 3 lineages. There's one colony was representing this lineage, and all the rest represented either this lineage or this lineage. And this is a smoking gun that this population of wild colonies went through a bottleneck, and the only bees that came out of that strong selection were ones that are able to survive with Varroa. And we looked also at the racial composition of the 32 wild colonies that we had, either back in 1977 or the modern ones, 2011. And you can see their racial composition is pretty much the same. They're originally, they were mostly a Ligustica carnica mix. Some of the old black bee, Apis mellifera mellifera, was a component. And there was some of the Caucasica or Caucasian bee, which was, when I was a boy, that was quite popular. So that was that. And that's what we have, that's what we found in 2011 as well. Though we did get some indications of the Scutellata, which had, of course, reached the Southwest and Florida by then. And it's probably some of those that got into the genetics, because there are migratory beekeepers that bring their bees up from Florida in the spring up to our area to pollinate apples. And there's this curious bit of genetics of another subrace or subspecies, Yemenitica, which is also African. It's part of the African stock. Pretty neat what genetics can tell us. Now, so it was clear from the genetic analysis that these bees went through strong natural selection. And the question is, what effect did that have on the bee's physiology and behavior? Well, I only have time to tell you about a few things, but it is favored workers that are good at killing the adult mites by biting them, biting down on their carapaces, and by biting off their legs, and also at disrupting their reproduction. I'll tell you how they do this. The earliest evidence we had was that it was the old and simple freeze-killed brood assay, where you take a comb of sealed brood. With sealed brood, you put a cylinder over it, and then you pour in about a half a cup of liquid nitrogen, and it freezes the brood and kills all the brood. And then you score, then you put it back in the hive, and you come back 48 hours later and you see how well did the colony clean out the freeze-killed brood, that is to say the brood that was damaged. And historically, people did these studies before Varroa, before there was any selection, and they found that it was only, the percent removed was about 30 to 40%. But bees that have been either selected by breeding or in the Arnott forest, it's almost 100%. So these bees that are out in the wild are very good at cleaning out diseased or damaged brood. They also have something else that they're doing, and I'm just starting to learn about this. I learned it from discoveries made in Europe, particularly in Norway. The bees have another behavior for controlling Varroa, and it's a behavior where they uncap a capped brood cell and then they recap it. And this evidently it disrupts the Varroa reproduction because it may frighten the mother mite. It certainly hinders the development of the immature mites because it's opening the cell, it's changing the humidity in the cell, and it disturbs the mating of her offspring. You know, she has one male and one son and a lot of daughters, and they mate together. But opening up that cell and then recapping it disrupts these processes. And you can tell if a cell has been uncapped and recapped is if you look at the cell and you see this sort of darker brown portion in the middle, that's where they've recapped the cell. So here's a normal capping of a cell, And here's one that's been uncapped and recapped. Again, there's a dark smear where the repair job has been done. And that's what we find in the wild. And I'm checking for this in the colonies that I've checked so far. And the way I check for this is I put up bait hives in the Arnott forest. I get bees living into movable frame hives, and then I can inspect their brood. The other thing that I'm finding is this very clear sign of the capping and recapping process. It's when you take out a frame of brood from your hive and you see pupae looking up at you. If you see purple-eyed pupae, these are cells that have been uncapped. And if it's at a glance, you see this, you know you've got bees that are doing this uncapping, recapping. I don't know if be curious to see if anybody out there has seen this phenomenon yet. It's eye-catching. So the answer to this question, are the wild colonies surviving thanks in part to having good genes? The answer is definitely yes, but that's not the whole story. But let me check in at this point, because getting this genetic story straight is really important. So let's pause now for questions.
We had one question from Tracy that you just answered. She was asking whether the bait hives just had just were a cavity or if they had frames, but you just said that they had removable frames, so you just answered it. And one participant said that they've seen a decent amount of the purple yellow eyes in the last few years.
Wow, that's great. That's good. That's very encouraging.
And another participant, actually Kit, just shared that she sees cappings opened, but she does not think that they are recapped after that.
Interesting. Okay. Yeah.
Any question, anyone? Or other comments?
I hope that, I think that, I guess I was just going to say in there's a little time here, just, I hope this is really encouraging that our bees, even though they had not evolved with Varroa, selection can very quickly give us bees that are resistant. I think one of our challenges is to figure out a way to get through to let the selection work. It's easy if you've got bees living out in the wild, but it's much harder if most of the bees in an area are living in beekeepers' hives, because then it's hard for beekeepers to not treat their bees. Should I go on?
I actually have had a couple of questions come in if you want to take them.
Okay, yeah, I think we've got time.
So one of the questions was, if you uncapped bees in the hives to do yourself to reproduce what you see in the wild, would the bees recap it, and would that mimic it?
I don't know. That's an interesting question. Or I really don't know. They may, our kind of uncap, the way we would uncap might not seem right to the bees, but it's an interesting question. To the best of my knowledge, nobody's tried that.
That's cool. If one of you wants to try, you can share back.
I would say get a small scalpel and be gentle, because it looks like it doesn't have to be the whole capping removed. Sometimes it's just chewing that opening, and the bees evidently, this is what they're finding in Norway. That period of uncapping may only be less than half a day. Just disrupts the mites quite quickly, apparently.
Interesting. That's good to know. We have a question about the races of bees and whether Linguistica is an old race, or whether it's unique to the Northeast in the United States.
Oh, no, it's not, because of the queen. The queen production process has just spread the genetics of all the introduced bees across the U.S. there are, that's certainly true for the Italian Ligustica and the Carniolan, the Carnica races. The Apis mellifera mellifera, it used to be you could, those were popular among beekeepers in the north because they are originally from Northern Europe. They're the bees that live north of the Alps and in Ireland and Scotland. And so those were popular among northern beekeepers, but I don't see them being advertised these days. So but they're clearly, that's a big component of the wild, of the genetics of the colonies in the wild, at least in the eastern US, in the northeast part of the US.
Thank you. Here's a naughty question that went with it, which is, what if we stopped all of us buying bees and try to catch wild swarms? Would we actually all have resistant bees and it would solve the problem of our colonies? I suppose you're going to get into colony size at this point around what you've seen versus what we do when we keep them.
Yeah, I do think that if every beekeeper in North America, which would be unrealistic because there's so many people that rely on the bees to make a living, if we went cold turkey, the problem would be gone in about 2 or 3 years. And I say that partly because I don't know if people were beekeeping when the previous disease came through. Which one was that? That was, Tracheal Mite. The first in the when was it? The 1980s, Tracheal Mite came in. We didn't have any treatment for it. People frutzed around with menthol, but we didn't have any treatment for it. And we experienced very heavy colony mortality. I experienced for 2 winters about 80% colony mortality in my bee yard, but now nobody cares about the tracheal mite. It's hard to find tracheal mites. So yeah, evolution, I think will give us a solution. And it may not, but it would come at a big cost, a transitional cost, because we just saw in the Arnott forest that they've got the ability to survive, but they went through a period, and I don't know how long it was. It might have been only 2 or 3 years where the population was knocked down to about 20%. So I think we have to find a different way, but that has been the pattern that has been seen in Norway and in Sweden, where they did experiments where they just took 150 colonies in Sweden, for example, out to an island, Gotland Island, and let the colonies go. And they had Varroa, and that 150 colonies over the next 4 years they're all killed off, I think, except for 10 of them. And then the population has rebuilt. So yes, that process will work. And there was a beekeeper in Norway commercial beekeeper that just had one apiary, set it aside, never treated it, and that's how he got his resistant stock.
That's really good to know.
Yeah, it is, and it's clear that it can work biologically. Whether it'll work economically and sociologically for humans is another question, of course.
Yes, the bees are very important to us in that way. There's one last question. I don't know if you want it, from Jeff, who has been actually working very hard in the Puget Sound area for breeding Varroa resistant queens, actually. And he says that he's been watching for flat-capped cells, and he's wondering if you've observed that behavior.
That might be. I don't look for that. I look for the discoloration to see if they're uncapping and recapping, but that maybe with your bees, maybe that's another trait. That's another way to look for it. But I haven't looked for that myself. But it might work. And the last thing I wanted to say is I just decided to go cold turkey like the beekeeper in Norway, and I haven't treated my bees for 5 years. And I lose about 30% of the colonies each year, but the other 70%, they do just fine. So we have the variation in our bees, and it doesn't look like the losses are astronomical, not as bad as one might think. So just a side point there. What about lifestyle? How regarding these wild colonies surviving? Is good lifestyle a matter? Yes, wild colonies enjoy at least 20 advantages over managed colonies in dealing with Varroa and other issues in life. One is the wild colonies are widely spaced. On average, they're a half mile apart. Instead of, and this is one of my old apiaries, instead of 0.5 feet apart. So this means there's less spread of pathogens and parasites. And we know now that with Varroa, the way Varroa really gets around is when, through the process of robbing, when a colony collapses and neighboring colonies go in and rob it out, the Varroa climb on the robbers and get transmitted into the colony that's doing the robbing. We've done experiments on that, and it's not a pretty picture. Even colonies that are 300 meters away will rob from a colony, and bring home the mites. But spacing the colonies a half mile apart, it really helps these wild colonies survive and avoid picking up cross-contaminating. Another thing about lifestyle is these small nest cavities they're living in. This makes them much poorer hosts for Varroa mites, partly because they have fewer cells of brood, but most importantly because they have more breaks in the brood rearing from swarming. I don't know if I put that into this talk, so I'll emphasize that now. Swarming is a hugely valuable aspect of how the wild colonies are coping with Varroa. They swarm, virtually every colony swarms every year. It has a brood break, and that just really knocks down the mite population in the colony, partly because a lot of the mites get exported in the swarm, and partly because there is a period of time when there's no capped brood in the colony, when a colony swarms, and then the mites are exposed to the killing of them by the worker bees. So small cavities and thus swarming really helps the colonies in the wild. Here's an example where I did an experiment just to see how this exactly works. I set up two groups of 12 colonies in the same location. They were both by this old storage building. In one group of 12, the colonies were kept in a single deep 10-frame hive body. In the other group, they were kept in what is this usual arrangement in central New York for commercial beekeeping, 4 deep hive bodies, with the bottom 2 the brood nest and the top 2 honey supers. And all the colonies were started out with sister queens. So they were genetically as close as we could make them. And no surprise, in this group of the 12 small hive colonies, 12 of them swarmed in the summer of 2013. That was their first summer where they were set up. Whereas in this group, there were only 2 out of the 12 swarmed, which isn't surprising either. There were big hives, they didn't get crowded, they didn't swarm. And here's what we've found about the mite counts. These are counts of mites per 100 bees. So these are like percentages at the end of the summer. In the large hive, it was 6.2 mites per 100 bees. So it was 6%. In the small hives, it was 1%, 1.1 mites per 100 bees at the end of the summer. And none of these bees in the small hives, we didn't see any bees with deformed wing. And 33% of the colonies in the large hive treatment group had deformed wing bees. So a big difference. But the biggest difference was in survival. In the large hive group, only 2 out of 12 survived the following winter, whereas in the small hive group, 10 out of 12 survived the following winter. Now, you might wonder, well, how was it that 2 of these colonies survived, even though they were in these big colonies which didn't swarm and had lots of mites? Well, it turns out one of these colonies, one of the survivors, was an exceedingly mean colony, very, very defensive. I should say defensive, not mean. So defensive that I never wanted to go into that colony, and it knew all about how to control Varroa mites. The Varroa mite count stayed very low in that colony. And so that was another way for these colonies to survive, to have the really be just very defensive, and including defensive against Varroa mites. And that colony, I brought it back out of the experiment, and it was so defensive. I didn't want to go into that hive, and I just parked it up in a corner of a field for 4 years, and it lived for another 4 years, all on its own, no treatments. Pretty tough bees. A third thing about lifestyle is the wild colonies use a lot of propolis. I don't know if you've never seen bees coming home with loads of propolis. Here's a nice little photograph. It's really rather pretty stuff. They bring it home, it's shiny, and they bring it in the nest, they go up to wherever the propolis is being used, and they just stand around until the bees that are working the propolis chew it off their legs, bits of it off their legs, and then apply it. And here's what I've showed already is how they can, even in the wild, they'll coat everything, even the floor of the nest cavity. And work by Marla Spivak at the University of Minnesota, she's done very, very carefully controlled experiments, show that having this propolis envelope all around the nest lowers the levels of activity of the bees' immunity genes. So they're not as, they don't have to work as hard to fight infections. And that also translates into better nutritional status of the young nurse bees. So that those young bees that are producing more bees are healthier if they have lots of propolis on the walls and floor of the nest. And in the wild, they do that. What we do with our standard hives, of course, is they're built of planed lumber. And so we just unintentionally, we've removed one of the most important stimuli for the collection of propolis, that is rough surfaces. But I'll just say you can fix that by putting in screen on the side of your hives, or companies even sell Propolis Collection screens. You can staple those on the walls of your hives to stimulate propolis collection and application. Another difference is between the wild lifestyle differences. The wild colonies occupy these well-insulated cavities. Here's and our managed colonies are just these thin-walled, very poorly insulated boxes. And here's an example to look at the difference between the insulation between a colony living in a hollow tree and in a hive. I cut with a colleague into a sugar maple tree. We cut a shape the size of this box here, and we were mimicking a wild colony's nest over here, and we built a box over here that mimicked the size of the shape of this wild colony, size and shape of it. And here's the tree with the door put back in place, and here's our box. And then we didn't heat these boxes. We just wanted to compare the stability of the temperatures inside them. And here's what we find. In the black line is the ambient temperature. This is just several days in April. It's chilly time of the year. This is the ambient, this black line. It jumps all over, goes up, goes down, etc. And here's the orange line is for the box, and it really basically jumps around virtually as much as the ambient, the outside temperature. But here's the tree. The temperature inside that tree cavity is very stable. It's no higher or lower on average, but it's much more stable. In the spring, as things start to warm up, it stays a little cooler in there. But the key thing is it's a very stable situation for bees living in the wild. Can't say for sure, but that almost certainly is less thermal stress, not having to deal with really high temperatures and then really low temperatures. I'm running out of my time, so I'm going to just go into these take-home lessons now to summarize the points I've made. Some wild colonies of European honeybees can survive without chemical treatments for Varroa. That's what I'm finding already with my bees. And I should have explained that I'm getting my bees for my apiary by putting up bait hives and capturing swarms from wild colonies. I put these bait hives up in remote places away from beekeepers. And the second main point is that how we keep bees is not how they live in nature and is not entirely bee-friendly. When you step back and think about it, almost everything that we do in beekeeping is good for beekeepers, but not necessarily good for bees. We crowd the colonies into apiaries for our convenience, but that facilitates the spread of disease. We house the colonies in large hives to get us greater honey production, but it creates less swarming and poor colony health, especially now that we have Varroa. We discourage colonies from coating their nest walls with propolis by putting them in hives that are smoothly planed on the inside surfaces. and this leads to weaker natural defenses against microbial infections. We treat the colonies with miticide. That's good in the short term, but it hinders natural selection for suppression of mite-resistant stock, bees able to suppress the mites' reproduction. And we discourage colonies from producing drone comb. And the consequence of that is it means that there's less drone rearing. And again, it hinders natural selection for resistant stock, because it means even if you have a colony that's very resistant to Varroa, if it can't rear many drones, it can't express its genetics. It can't spread its good genes very effectively. So my advice for miticide-free beekeeping, if you want to try it, give it a try. And it really helps to do this in a group, but you can do it on your own if you're remote. Rear queens from your colonies with the lowest mite counts and best survival. You don't have to, I bet you'll have variation, just as I keep finding variation in my bees. Some colonies keep the mites low all on their own and survive well. So you breed from those, and that's what the beekeeper in Norway did, and others have done, the beekeepers in Wales and the beekeepers in Ireland. All of these are groups where they've just given up on treating and work with their own stock. Second advice is make splits using the queens, worker bees, and broods of your strong mite-resistant colony. Just let the donor colony requeen itself. It's a simple. You don't have to be a queen breeder. You just make splits and let the bees rear from your good colonies, and let bees rear the queens on their own. Give your survivor colonies, once you've got some good survivor colonies, once you treat and they can survive, make sure they've got a frame of drone comb per hive body so they can spread their genes. And if you must buy queens, use only queen producers who do not treat their colonies with miticides. There are not very many of them, but there are some. There's one here in Vermont, back east here in Vermont, Kirk Webster. I've tested his stock, and his bees, they kill them, are under control. At the same time, I tested colonies from Conan queens and from Oliveira's queens, and those colonies are absolutely helpless with respect to the mites. So those queens are not to be recommended, and that's, of course, they're not selecting. And so that's my advice for miticide-free beekeeping. I've covered a lot of ground, but as was mentioned before I gave my talk, I did write a book on this subject. It's called The Lives of Bees, and it goes through in more detail the things that I've talked about tonight, what I call the untold story of the honeybee in the wild. I think it's a good read. I hope it's certainly, I think whether or not you want to go for treatment-free beekeeping or not, I think it's always good to know what the natural history of your organism is. And that's what this book is about. It talks about how bees live in the wild. So I think that's my last slide. So I guess if you'd like, we can do more questions. I'd be happy to do so.
That sounds great. We actually had one that came as you were speaking, which was that, let's assume you treat, and this was a thymol treatment. The participant is wondering if that strong smell of thymol can hinder bees' capacities to communicate with each other, their pheromone communication.
Yeah, I don't know. I suspect it does, and that's one of the many things that's unknown about our treatments. That thymol is really potent stuff. I mean, the aroma is very heavy.
Thanks. I actually have an EpiGuard on 3 of my hives right now, mostly to kill the chalk brood, and one of them refuses to go into the box that has it, that the EpiGuard is on top of. So they're really reacting to it. They hate it.
Yeah, it is. Yeah, it is. It's sort of amazing that the bees can survive with that much material in the atmosphere that they're breathing.
That's true. Does anyone else have any last questions?
Maybe I could ask a question. of you guys. Does this seem, is there anybody there for whom it would be possible for them to try this, to have an apiary where they just let it go and see what happens? It doesn't need to be a big investment. It might only be 5 colonies. It'd be nice if it were 10, but and it would ideally be a person who's not amidst a lot of other beekeepers. But I know you've got a big, you've got a big state out there. You've got a lot of land, probably a lot of remote places. I don't know. Wonder if it's possible for somebody just to try this approach.
I actually I was at a point where, because I'm a younger beekeeper, I was a lot in correspondence with Kathy throughout the season. She's been keeping bees for a very long time. I'm very isolated where I am. The first beekeeper is probably 10 miles away minimum. So I am aiming for that. What I think I need to do is buy queens that are, in theory at least, mite resistant, and then just monitor and see what happens by the end of the season.
If you want to buy queens that have very good mite resistance, the person I know and whose stock I have tested is named Kirk Webster. Kirk Webster. And he's not real, I don't think he has a website, but I think somebody actually built him a website. And I've tested his bees. If you send me an email, I'll send you a copy of the article I wrote for American Bee Journal, where I tested his bees and compared them with Oliveira's queens, and I compared them with my wild queen colonies. And his bees are the best at dealing with Varroa. And he knew many years ago that he wanted to see if he could get bees that could survive without being chemically treated. And he's got that stock. And he uses his stock not just to make more queens, but he runs a whole big honey operation as well. So his bees know how to do it. And he breeds them in Vermont. He mates them up in the mountains of Vermont, where he brings up, so they mate only with drones from his mite resistant stock. So he has a good product.
That's cool. Thank you. We have one question from a participant, which is, from all of what you said, does it make sense then for us to make more splits throughout the year to imitate swarms and to keep smaller colonies?
Yeah, you can, even with a colony that doesn't have a great queen, a queen with the right genetics. If you mimic swarming by making splits, you will make a huge, you'll gain a lot of control of the mites. And the key there is to make the split, to go to a colony and split it, take away the queen and several frames of bees and brood, put it in a nuke box, take it away, and then, but don't put a queen back in the colony that you dequeened. Let the bees go through their emergency queen rearing so they have a break in their brood rearing. That in itself is actually a pretty good way to control Varroa without using chemicals. It will harm your honey production, but so there is that trade-off, but it's a way to do it without having to give them chemicals. And along the way, you will be selecting, and then you'll be finding some colonies can do very well in that situation, others don't do so well. So you just let the ones that don't do well die out, and you build your numbers back up from the ones that do well. That's at least that's what I'm doing now. And as I say, I lose 30% of my colonies each year, but the ones that survive, they come through they're very healthy colonies. They come out in the spring. They're great, in good shape, no mites. It's really a pleasure to work with. And the reason I'm getting 30% each year is I'm capturing swarms, and some of those swarms come from colonies that don't have the right genetics. So I keep unintentionally, I keep bringing in lousy stock, but I don't know whether it's good or bad. So I just have to take what I get. And the bees themselves will tell me whether they're good or bad by the end of the summer.
That makes sense. I can actually attest to the fact that brood breaks really, really work. I have four colonies, and one of them got split twice. Actually, this year they were wanting to replace their queens all the time, and by the end, I counted the mites about 2 weeks ago in my hives, and I had only one that needed treatment. The others were below the 2%. And I don't know that it's particularly good stock. I don't think I didn't buy queens that were particularly resistant to begin with, but those brood breaks really made a difference.
Yeah, that's right. Because it really exposes, it means your bees are actually killing the adult, are killing the varroa. Because the way the brood break helps is it means the mites can't reproduce and they can't hide.
I wanted to make a comment because I sell about 25 nukes every year. I have a brood break in every single hive that I have. The ones that I don't think are all that great, I keep the bees for honey production, but I have specific ones that I breed from. I have right now in my apiary, I have 2 3-year-old queens that are just fantastic. And I also breed for Propolis Production. And in taking my master's class at the University of Montana, my paper was actually on Propolis Production in hives. And we tested out putting propolis traps stapled to the inside, window screens stapled to the inside. One side was the control, and the other side, I used a drill, and I don't know what it's called. It's called the thing on the drill. I'm not at all mechanical, but and gouge the sides of the boxes. And the Propolis Production was absolutely amazing. And those hives that are in boxes like that are outliving doing all the rest of the hives.
Yeah, yeah.
Trying to follow Marla Spivak. I took a class from her in, I think, 2004 or 5, and had a big discussion about propolis because I really liked it. I thought it was a great medicine, and I wanted to have bees that produced more of it. And she said it's great for the bees too. And I just have been thinking about that and have always selected for bees that propolize the heck out of everything. Harder to work, but I think that's really well worth it. And the cost of doing the propolis traps, if you have a couple of hives, it's not bad, but if you've got 50 hives, it's cost prohibitive. So the gouging the side with this little drill with all the wiry that goes around in a circle and gouging the side of the hives really worked well. But the initial cost is just the little attachments to the drill.
Yeah, yeah, that's great. Yeah, yeah. Thank you for sharing that. It doesn't.
Yeah.
That's you should write that up for the American Bee Journal. Share your experience, pass it on. Yeah,
Well, I could do that.
that would be good. Yeah.
I feel like I should jump in and do a little plug for the breeding I've been trying to do locally here. As a takeaway for people, it's not always best just to let your bees die in a city location because we are so close and we create those mite bombs that will get robbed out and then will hurt the robust hives that do have the traits we want. What we want to see people doing is to test their mite levels, and if they have high mite levels, replace those queens. And go find someone, and I'm sure Vermont, you can get great queens, but find someone that's breeding queens with treatment-free qualities that are adapted to the Northwest, that know how to deal with our high humidity winter climate that doesn't get very cold, but it stays wet all winter and misty and rainy. Those are the queens, those are the bees we want here. And you don't have to let your hives die necessarily to know that they have high mite counts, they're not going to survive. So swap your queen out, and if there's not time for that season, treat them, get them through the season, and then change your queens on the next spring.
You're here, Jeff. Thank you for pointing that out. I don't think I made that point clear that, yeah, you really don't want to, you don't want to let your colonies die in the summer from high mite counts because they've become these mite bomb
Right.
robber lures. That's a disaster even for colonies that have resistance if their foragers bring back loads of mites. And I take your point too that, yeah, it sounds like your conditions are moisture is so much more an issue that, yeah, you've got to get bees from Vermont are probably not going to be appropriate.
Thanks.
Yeah, you're welcome. I've heard I've got some beekeepers in remote parts of Washington have told me about these colonies they find way out on some peninsulas and stuff like that. Is anybody here in touch with those beekeepers? And they say they're very black bees and things like that. Has anybody heard tales or stories or reports of those wild colonies?
Tom, I have a cabin out on Hood Canal, which is on the peninsula.
Yeah.
And I've got anywhere between 5 and 10 colonies that I run out there. It's pretty remote. And those bees are really black as well. And they're very defensive too. I wouldn't want to have them in town, but they're rock solid because they get neglected by me out there. And half the time I don't treat them and yet they live and they're really good honey producers too. But they are very black and they're large bees too. They won't fit through, the winter bees won't fit through a mouse guard.
Interesting. Wow, that's pretty cool. That's really cool. Sounds like you've got several colonies of the sort that I had in my experiment. Really, very
Maybe.
defensive. And they could just, if they saw Varroa mite, it was like toast for the Varroa mite, apparently.
Yeah, the mite counts are usually under 2% out there. And like I said, last year, I got one batch of Apogard on. I usually treat with Apogard in the fall in town just because of the mite bombs. But last year they got one treatment of Apogard and then I forgot about them and didn't get back over there. And they did just fine.
Thank you. That's really neat.
Sure.
Did you source them from swarms that you picked up over there, or?
No, so well, I captured one swarm last year, but I'm pretty sure it was from my own colony. I originally sourced the bees from Lori Miller that were over there, and they were dark feral bees that she had captured out on Mount Rainier. And then I've just let them naturally breed out there. But they are pretty defensive. Like I said, I was going to sell some nukes, but you cannot go out into, and I have them in a dog kennel because there's bears out there with hot wire around them. And you can walk by outside the dog kennel all you want, but don't go inside without your suit and smoke or they will seriously pelt you.
Yep. That said, that trait doesn't have, that it is a marker of colonies that are very defensive, and that defensiveness apparently applies to Varroa. But the beavers in Norway and in Wales especially, they are not, this is a group, particularly, I know the ones in Wales the best. It was a group of beekeepers on a peninsula that just said, no, we're gonna do an experiment. We're all gonna just stop. And they've got bees that are not very aggressive, but they survive without the treatments as well. So we don't always have that, but that high level of aggressiveness, but evidently it does help, but it's not necessary.
Yeah, we don't see the aggression. You can work, at least the treatment-free colonies that we're sourcing here, you can work glove-free.
Yeah, wow, good. That's good. Yeah, I'd have to say the colonies that I have were in my apiary. Well, I just have one apiary now, and it only has a dozen colonies in it. I don't think I have any really nasty bees in there, but I've got lots of colonies, at least 3/4 of them that control the mites very effectively. And that might reflect different ways of defending against Varroa. Who knows? We're not sure.
Hmm.
A lot of food for thought there.
Yeah,
Yeah.
it's such a unique situation that you're able to experience. And you're living in a rural area, so you can allow swarms and not be concerned about where they wind up. And
That's a really important point. Yep, that's right. And I try to keep that in mind, that what I'm saying is not applicable to a lot of beekeepers, anybody that's in an urban situation where swarming is a problem. Mhm.
I always
yeah,
find those aggressive bees are more trouble than they're worth, and they don't produce much honey, and they're not very mite resistant. At least the ones I find in the city, they're just mean, and they waste all their energy on being mean.
There's a lot-
Tom, do you think that doing like a Terranoff split or shook swarm, the kind of phony splits, do you think that you would have the same results with doing splits like that as opposed to bees that are building queen cells and swarming naturally?
Yeah, yeah, I'm not familiar with all the different ways of doing splits, I'll have to say, but any manner of splitting which creates a brood break.
It's the brood break that is the thing.
Right.
Yeah,
Yep.
thank
That's-
you.
That's
So
my
I think if you were to do that correctly, you would move the queen resources, but no larvae, no brood, to the new hive. And then the old hive that's raising a new queen would keep all the brood and capped cells. And then each hive gets a complete reset because you have ferric mites in the queen right side with no brood. So they can, knock that down. And then the other side won't have a queen to lay, so they'll have a break too.
Yeah, that's a very nice way of putting it. Yep, very good. Thank you. That does express the logic very nicely. Queen but no brood, brood but no queen. Tough on mites.
hmm. So the other thing is, doing splits, which is such a good solution, also creates so many hives. Then what do you do? I mean, happy bees multiply, and you have a lot of bees.
Yeah, you can sell them. Nukes are pretty profitable where I am. Not that I
Yeah.
do it, but I know a lot of beekeepers, that's a big part of their income, it's making nukes, selling nukes.
Yeah, that's here
I'm not
too.
getting rich off it, but I always feel like when I send a nuke out, number one, I also breed for gentle bees. I work my bees in just whatever I'm wearing. I don't use a veil or gloves or anything. And I feel like they can go into any neighborhood. A beginning beekeeper is going to have an easier time working them. And I like the idea that I'm sending good bees out into the neighborhoods as opposed to just any old nuke,
Yeah.
which is kind of what you get when you get California nukes.
Mhm.
They're just taking the bees right off of the almond pollination and creating the nukes there, just any which way, and then selling them to us.
Mhm.
It's a different world. Yep.
Yeah, here I get, I know that my bees have overwintered for years, and they've got some really great traits, and I feel really good when I send them, like I said, when I send them out into the neighborhoods.
Mhm. Yep.
Well, it's been wonderful to have you, and
Thank you.
it's really, really a pleasure.
My pleasure too. It's given me a lot. It's hearing what you talk about your beekeeping and the moisture issues and stuff has really given me food for thought about Oh, yeah, that's a different world. Moisture is a different, it can be a serious matter in places. Got to keep that in mind. Good. Good.
Yeah, and our bees drown from the rain inside their hives if we don't set it up so that they have, so that there's something to absorb the moisture. Yeah,
Yeah,
we learned that right in the very beginning.
yeah, that's different. We and that's true even if it's a well-insulated hive, so that there's a, yeah.
Well, the insulation helps for sure. That's and tipping the hive forward is critical.
Yeah.
But yeah, the water, we just got to keep that water from collecting inside, because that heat creates condensation. You know, the bees are creating so much heat and there's so much humidity in the air from the rain.
Yeah.
That's a cycle we have to protect them from.
We find that, and this isn't my work, it's a beekeeper in New Jersey. He's got this neat technique where he puts, instead of a wooden inner cover in the hive, he puts a sheet of plexiglass, and then he can open the hive up. He will put a foam board over the plexiglass sheet, but it lets him score a colony in terms of its the moisture, because he can, the plexiglass will either be covered with droplets or not. And what he finds is that if the colony's living in a well-insulated hive with a foam board over the inner cover, like you were talking about, there's such a warm pocket in the top of the hive where the bees are. The surfaces where the bees are nice and warm, and the condensation doesn't occur there. It occurs below them. So I think that's what happens in the wild too. They're up in those thick trees. They're well insulated, and they don't have top entrances. But I think all the surfaces are above the dew point, and so they don't have water on them. So it's something to think about. And so, especially where you are, where you've got lots of moisture problems.
So the moisture is below the bees.
I don't know what, my phone just went off.
Yeah, the condensation occurs below the bees because in the well-insulated hives, on the walls and the top, there's no surfaces that are cool enough for water to condense on. And the other trick he does is he avoids the top entrance, so the heat stays in that upper warm pocket of the hive.
That's very interesting.
It'd be interesting to do an experiment on that. Now he's working in New Jersey and he doesn't have your humidity, so it's a different world. But it's like, I think he's got an article coming out in one of the bee magazines where he shows these just dramatically different photos between insulation or without, for a colony in where he lives. Even in New Jersey, if it's not well insulated, there's lots of condensation. But if the hive's well insulated, then there's up where the bees are in the winter, it's warm and the surfaces are not cool enough for the water to condense on them. And
I wanted to also say that one of my first mentors, Serge Lebesque, he has written for Bee Culture magazine, and he is in Sonoma County where I started beekeeping, told me about follower boards. And so I have all mediums now. Since I relocated, I get to start over. And so I have all mediums with follower boards in position 1 and 10. That means I'm really running an 8-frame and a 10-frame box. But what happens with the condensation, it goes up on the top, and then with the follower boards, it creates a convection that pulls the dampness to the outside of the follower board and the inside of the hive body, and it never touches the bees. And when I open things up in spring, there'll be mold in those areas, but never on the comb. So that's really helped. And then I use rigid insulation on 3 sides, because where I'm in a convergence zone, and I have crazy weather here, and the water will wick sideways and it'll go in between the hive boxes. So by putting the insulation on, not only am I trying to make them more stable, but I'm also eliminating that water coming in. I leave the front open, I leave robbing screens on all year round, and I do keep a top entrance in addition to using a moisture board and a moisture quilt. So trying to keep those bees alive.
Yeah, wow, that's there's a lot of It's a very complicated situation you have. I want, wish I had firsthand experience with it. Sounds
The important thing with the follower boards is to make it like a chimney. They've got to go one on top of another all the way up. If you've got 3 boxes, all 3 boxes have to have them to have that work like that.
Yeah.
But it's quite nice. And then when you're working the hives, you take out a follower board and you've got all sorts of room to manipulate without crushing any bees. I just really stuck with it because it really seemed to work and be good for me and the bees.
How thick are those boards, those follower boards?
They're all I do is I take a piece of 1/2-inch plywood and I cut them to the size of the frame without the top bar. And then I buy top bars, 3 screws into the plywood, and I've got a follower board, and it creates bee space on either side. The ones that I'm seeing being sold in the catalogs are just solid, no bee space. And so if you try to move it over out of the way, you're crushing the bees against the side of the box. So I'm always trying to, initiate new beekeepers to follower boards and tell them how I do it so that they make ones that are good for the bees and not bad for the bees.
Yeah, good.
Yeah, I'm a bee nut.
I think I'm starting to fade at this end. So
Okay, Tom, thank
Yeah.
Good
you.
night.
It's
but
been wonderful to have you. I know a lot of people will listen to the recording too. That's going to be a wonderful asset.
well, you guys are a great group. It's been really a pleasure to meet you and share what I've heard and learn from you. So thank you very much for inviting me.
Thank you for joining us. It was very special.
Thank you.
Good night.